Okay, I thank the organizers for having me and you guys for all coming. My name is Kevin Alseri, and I'm from the Loci Lab at University of Wisconsin, and I'll be following the theme of trying to have informatics solutions for imaging, with this talk really focusing on what we call bioimage informatics, which I'll explain what we mean by that. That'll be a twist on terminology you've heard before, and then also the particular challenge we have that all our data is multidimensional, and going through that challenge. And I hope to introduce my work, but also two of my colleagues that will be speaking after me, kind of introducing some of the concepts that they're working on as well. So my lab's main mission is to develop new optical instrumentation and new software for dealing with multidimensional data. I'm focusing on software today, but I'm also going to highlight the instrumentation a little bit because that drives our opportunities and our challenges. And I'll be going through quite a few websites, but most of what I'm talking about is all available on our Loci website. So the term infrastructure has been around for a long time, and it's been obviously used to describe commercial infrastructure, public roads, and so on. The cyber infrastructure has basically been a new term over the last maybe 15 years of trying to describe what you need to actually do informatics in academia and in the commercial industry. And traditionally, when one defines cyber infrastructure, I got this from Wikipedia, you can see all the various categories of cyber infrastructure. And if you look at our needs, you can see that we have an example of each one of those. So when we talk about bioinformatics, we are talking about data acquisition, so software for that. Also, it can include hardware like FPGAs and DSPs and other types of hardware. It includes data storage, file server and RAID, data management, databases, and then data integration and trying to deal with how you can make the correlation, the result, interpreting it, going from genotype to phenotype, for example. The data mining, drug discovery, linking in with other database systems, including the brain Allen database, and then data visualization, cell tracking, 3D rendering, and other kinds of systems. So I'm giving some examples of these, but these are the core areas that we, in this field of bioinformatics. And bioinformatics is a subset of bioinformatics. It's closely linked. Again, if you think about what bioinformatics is in genomics, it includes the development of new algorithms and stat approaches, analysis and interpretation of various types of data, and development implementing tools that allow you to manage that data. Again, we have all these same opportunities in imaging, and so there's a lot of parallels. And for the most of my talk, I'm going to talk about the differences and where the unique challenges in imaging. But you can see a lot in common between the two topics. So what exactly is bioinformatics? Well, it's a subset of computational biology and bioinformatics. It's about trying to use computational techniques to get your data, analyze it, and figure out the core information that's there. It's trying to do it on a large scale and high throughput when necessary and when possible. And the big challenges and the opportunity is that it's dealing with heterogeneous imaging. It's also dealing with metadata. You'll be hearing, I think, a few talks about ontology and metadata and the challenges that, again, the opportunities. And with every challenge, there is an opportunity. And then novel image processing methods, ways to actually extract this data. And then this field is in super rapid growth, in large part due to just some amazing new tools that have been developed that I'll briefly talk about, but also parallel technologies like the genome sequencing effort. And in general, what I'm going to be focusing on, and the field's been focusing on, is light microscopy, biophotonics, in particular high-resolution methods that allow you to look at cell resolution. That largely is due to the biology, really trying to understand things at the cell resolution, and the understanding that events like cancer progression really can be understood better, and drug discovery to do at the cell level. However, there are opportunities for other